I would like to thank the organizers for giving me this chance to share some of the findings. We have been working for years with this audience. And as Dr. Masood started saying that it's a serious problem, we all know that it is. And these 3 days, I think we're going to talk about this serious problem. So why not to start on a light note? So before I start talking, I'd like to share a joke. So when in the, you know that I'm coming from Pennsylvania, a cold place. So in the early days, when the Weather Channel started, and in America, there were a lot of Native Americans, or Indians, so-called, not like me. So when the Weather Channel started, the tribes, they said, why not to take advantage of the new technology? So the tribe chief appointed one person, asked that every week you go and call them, find out how the weather is going to be this year so that we can be prepared. So when fall comes, the first person calls first week, how is the weather going to be? The answer is it's going to be cold. The next week, he calls, and the answer is it's going to be very cold. Next week, it's going to be very, very cold. And next week, extremely cold. And finally, it's going to be historically cold. So the chief keep asking, collect more and more roots, more and more roots. Ultimately, the guy asks, why is it going to be historically cold this year? The answer is because Indians are collecting historically high level of foods this year. So the bottom line is that what we're going to discuss in these 3 days is not going to be based on hypothetical situation that what the answer I want to know is coming not based on the actual facts, rather just seeing that somebody else is doing something. So this is what going to be. And that again comes to Dr. Masood's observation that we have to be very, very careful. But jokes apart, now come back to the serious science. But I can assure you that what I'm going to talk is based on facts, and so is the rest of the group here. So breast cancer can be divided into 2 major groups. One is based on the positive expression of estrogen receptor, progesterone receptor, and HER2 receptor, and they can be treated using hormonal treatments. And then there is another class which is negative for the expression of these receptors, and there is no specific target as of now. And we call them triple negative breast cancer, and I'm using term TNBC for triple negative breast cancer. And this kind of cancer accounts for 10 to 20% of breast cancers, and they are very aggressive, and the relapse rate is very high compared to receptor positive ones. And as I said that there is no specific target therapy for TNBC, so we have to rely on radiation, surgery, and chemotherapy. So the standard course of treatment is very aggressive surgery and radiation therapy combined with adjuvant or neoadjuvant. Both kind of chemotherapy we use for TNBC. And taxane is usually used for chemotherapy for these kind of cancers in combination with other kind of chemotherapies. And as I mentioned that there is no specific target, and that's where people are trying to find out specific targets, and we are working on that too. And I'm going to share some things which we have learned so far. So one of the targets recently has been found that glucocorticoids, and depending upon the level of glucocorticoid receptor expression, the triple negative breast cancers have been shown to express much higher level of glucocorticoid receptor, And a recent study came up which showed that in fact in triple negative breast cancer, uh, the higher level of glucocorticoid receptor expression leads to anti-apoptotic effects. And that could be one of the causes for the high level of oncogenic activity in triple negative breast cancer. And in fact, a group from Chicago, they showed that if you use anti-glucocorticoids, for example, RU486, that acts as an antagonist for glucocorticoid receptor, can be used to block those anti-apoptotic effects. And in fact, that can be correlated with several anti-apoptotic genes. So these results suggested that, in fact, blocking glucocorticoid receptor activity may be a good target for TNBC. Another study came up very recently which showed that a coactivator protein, steroid receptor coactivator 1, SRC1, that acts as a coactivator for many steroid hormone receptors, including estrogen receptor, progesterone, and glucocorticoid, to name a few. And what it does, it binds through an LXXLL motif, and this binds to the ligand binding domain of the receptor that I'll show in next few slides how it works. And so when they use this peptide, designed a peptide to block the site that cannot interact with steroid hormone receptors, they found that in fact it helps to lower down the rate of proliferation of triple negative breast cancer. And they did that in the estrogen receptor positive as well. And they concluded that in fact the effects are not through estrogen receptor. That raises a very good question. So if SRC1 acts as a coactivator for estrogen receptor as well as glucocorticoid receptors, could it be acting as a coactivator for the glucocorticoid receptor in TNBC, and that's how we get the proliferative effect due to glucocorticoid receptor. So if that's so, if we could block the interaction of glucocorticoid receptor with SRC1, we might be able to use it as a therapeutic target for triple negative breast cancer. So in general, when we talk about targeting steroid hormone receptors for any kind of endocrine-based cancer treatment, the first thing comes to mind is how to have cell or tissue specific effects. Because we know that when we use antagonists to block receptor activity, in many cases, for example, if we talk about estrogen positive ones in breast cancer, when we use tamoxifen, that is very commonly used to treat them. In fact, tamoxifen is good as an antagonist in breast tissue, but it acts as agonist in many other tissues, including ovarian. So that could potentiate the tumor formation elsewhere while it's protecting. So you don't want to have a situation where there are such a huge side effects. And in general, even if we think about steroids, one thing worries to everybody to use them is side effects. And this is the main reason, because we cannot control cell or tissue specific activity by using agonist or antagonist, because on one hand they could work in one cell tissue as agonist, another one an antagonist, or other way around. And in fact, for last several years, one approach has been to use selective steroid receptor modulators to treat any kind of endocrine-based cancer. So they can be manipulated in tissue-based therapies where you can have partial agonist or antagonist activity. To a certain extent, it has been successful, but not completely. So now coming back to what I was talking about, the glucocorticoid receptor, this is a topological diagram of human glucocorticoid receptor, which consists of 777 amino acids and is divided into three domains: ligand binding domain, LBD, on the far right hand side, or C-terminal. In the middle you see the DNA binding domain, and rest is N-terminal domain. And within the N-terminal domain you see AF1, which is activation function 1 domain, and there is another one activation domain, which is AF2 on the far C-terminal. AF2 is ligand dependent, and AF2 is constitutively active. It can be active even without ligand. So the structure is known for the DNA binding domain. I'm not going to go into detail. And the structure of ligand binding domain is also known. And now, the structure of the N-terminal domain or the AF1 domain is not known. And this is not just for the glucocorticoid receptor, but all of the steroid hormone receptors. So why we always design antagonists when we look for blocking steroid or hormone activity in endocrine cancers? The reason is most likely this: because we have the structure of ligand binding domain, AF2 activity is here, activation function domain, which interacts with a lot of co-activators or co-repressors depending upon agonist or antagonist bound. So we want to find easy target, and pharmaceutical companies always go for easy targets because this is a very structured domain. We know how to manipulate the conformation of AF2, and based on the conformation of AF2, the co-activators or co-repressors bind. And like LXXLL motif I talked in the beginning is in fact the motif which binds to this domain. So if an agonist is bound, a co-activator will interact through LXXLL motif of co-activators. But if an antagonist is bound, it won't. And that's what we design all the drugs based on antagonist when we do, so that it can block the LXXLL motif interaction with the steroid receptors and thereby suppress the activation of target genes. For example, tamoxifen in breast cancer. When it comes to glucocorticoid receptor, 60 to 80% activity is governed by AF1 domain. Here, when we design an antagonist, it's just working through AF2. And that can tell the story why we not getting the whole spectrum of the receptor when we're targeting as a therapeutic molecule, because we are ignoring 60 to 80% activity. And if we do in that, of course, there are bound to be so many side effects. Plus, the efficacy is never going to be very high. Why we are knowing this very well for so many years, we're not doing that? The reason has been, as I mentioned here, this is easy molecule to work because the structure of this domain is known. We don't know much about the structure of this domain. So if we want to get the whole spectrum when targeting glucocorticoid receptor, or for that matter, in fact, all the steroid receptors, we must get the whole spectrum of the molecule. And to know that, we need to know how the activity of both AF1 and AF2 are governed. Another thing is both AF1 and AF2 work in a synergistic manner. So if you block in one activity, you are certainly doing it at your own risk. You have to block both the activities simultaneously. If not, we're never going to have the most effective treatment option. So why we don't know much about the structure of AF1 domain? Because this exists in recent years. We learned that it exists as an intrinsically disordered protein. Means it doesn't have any ordered structure. It's floppy, unstructured, whatever you want to use term. Well, for not too long ago, the idea was, or the thinking rather was, that if we have an structured region in a protein, probably forget about using or thinking even about a therapeutic target for this, because the concept we had in mind, so-called textbook model, was that protein has to have some kind of structure to design a molecule that can fit into. But recently we have found that in fact intrinsically disordered regions are very common in transcription factors. 70 to 80% of all the transcription factors, particularly their activation function domain, they are intrinsically disordered. And this is very, very true, in fact, with the transcription factors related to cancers, including p53 molecule. So large stretches of intrinsically disordered protein, about 2/3 of all the transcription factors involved in cancer have large stretches of intrinsically disordered domain. And now it has become one of the hot areas to target for screening new drugs. The beauty about intrinsically disordered proteins is that they look for a target molecule, usually protein or DNA. When they get those target molecules, they interact with them, undergo disorder-order transition, thereby they find a kind of structure. And that provides a platform for its interaction with so many other proteins. And we all know after genome that none of the genes work in isolation. They need partners. So when you have intrinsically disordered domain, you have flexibility to form any kind of conformation, millions, zillions of conformations in no time. So this gives them opportunity to remodel themselves depending upon the situation, whether they want to attract a partner or they want to displace a partner. So they work both ways, and that's why probably nature has deliberately created this kind of situation in proteins, which are mostly involved to work in a multi-complex proteins. So when we analyze the structure of AF1 domain of the glucocorticoid receptor, we made a recombinant protein of the AF1 protein, and as predicted, we did deuterium hydrogen exchange experiment. It's a very powerful tool to look for the structure. And what you do is exchange deuterium and hydrogen and apply it to mass spectroscopy, and virtually you get information about each and every amino acid present there, what kind of dynamics they are in. And the color indicates that if you have more red, it means those regions are highly exchangeable to deuterium hydrogen, and they'll be highly exchangeable when they are unstructured. If they are structured, it won't be easy to exchange because it will be difficult, just like proteases, to go and chop them. And you can see that most of the region, in fact, is highly disordered here. And then we did NMR, and again, that shows that it's highly unstructured. Why? Because this is showing the N15 label protein with the HSQC spectrum. And if this protein was structured, if you look the range from 8 to 6, they should be spread everywhere. That's indication of a highly globular ordered protein. But if they are confined to a very narrow range of 8 to 7 or something, that means the protein is mostly unstructured. And that's what it shows. So both these experiments, and in fact we have done tons of other experiments, and we have published extensively on this, all of them show that this is an unstructured region. And now this is the reason why we haven't been able to study so much, even though knowing that it's very important. And I already mentioned this, that they are important because of the flexibility, and they undergo disorder to order transition. And just like the AF1, I mentioned that they interact with with a lot of coactivators. In fact, AF1 does the same thing. And SRC1, the molecule I talked about, has been shown in TNBC, is known to interact with both AF1 and AF2 of the glucocorticoid receptor. So the question we ask, if that's so, is SRC1 in fact working as a coactivator? In triple negative breast cancer, where there is no estrogen receptor to take it as a coactivator, is glucocorticoid receptor replacing that and thereby leading to oncogenic activity. And the first thing why this came to our mind is that we looked for the progesterone receptor and we found the progesterone receptor AF1 is in fact same as glucocorticoid receptor, and that also have intrinsically disordered AF1 region. And when we use progesterone receptor, just like deuterium hydrogen exchange experiment I talked about in the previous slide, and we found that one very common protein from the basal transcription machinery, Tata box binding protein, which is in fact known to bind to most of the steroid hormone receptors, including estrogen, progesterone, and glucocorticoid. And it does bind only to the AF1 domain, not to the AF2 domain. This is very important. And when we bound TBP, Tata box binding protein, to the progesterone receptor and look for the conformational changes, we found that when the receptor is bound to an antagonist, it in fact leads to changes. I'm sorry, I'm showing here agonist bound. So when the receptor is bound to agonist, in fact, when TBP is bound to the receptor, not on the ligand binding F2, rather on the F1 far end towards the N-terminal, it in fact leads to conformational changes in the AF2 domain, in the ligand binding domain. I don't want to go into detail about the experimental conditions. I just want to emphasize that TBP binds to the AF1 domain, and when it's agonist bound, it alters the conformation of AF2 domain in the ligand binding. And look here, when it's antagonist bound, the same molecule, same receptor, it alters the structure in the AF1 domain where it is binding. So this gives a clue that this could be a molecule which is dictating in fact the effects of agonist versus antagonist, depending upon how it's expressed in a different cell or tissue. And we came up with this model very recently. We published this, that when you have agonist bound, you are changing the conformation of ligand binding domain here, and when you have an antagonist bound, you're changing the conformation of AF1 here. And we thought, why not to take advantage of this and see if we can target this as a therapeutic molecule for the glucocorticoid receptor in TNBC. The first question was to find out if the same thing works for the glucocorticoid receptor. So we came up with this hypothesis. Rather than using antagonists, we thought we need to target AF1, where the beauty is that we don't have to use a steroid molecule. At the same time, we can cover a larger activity, that is about 60 to 80%. If need be, we can in fact combine this with an antagonist like RU486 and cover the whole spectrum. So we came with the hypothesis that AF1 domain, which is unstructured, can bind to TBP and thereby undergoes disorder order transition, gets a kind of conformation that might attract SRC1, the coactivator molecule, and it can bind to the molecule and lead to target gene expression. So to test that, first thing was to find out if TBP does interact with the glucocorticoid receptor, and we created different constructs of glucocorticoid receptor because we were targeting AF1. So we deleted AF1 in one construct to see that if the TBP binds, is it binding through the AF1? And to test that, we use CB1 cells which are null in glucocorticoid receptor, so we don't have to worry about any endogenous effect. And we did FRET analysis, and what we found is this is a very powerful technique to study protein-protein interactions in cells, and we found that in fact TBP does bind to the AF1, and this is before photo bleach and after photo bleach. If you see the red color, that is indication of protein binding. And in fact it does, When we take off or deleted AF1, it does not. So that, and this is just a control to show that the experiments were good. So that showed that TBP can bind to the glucocorticoid receptor AF1 domain. That's good. And then does it, and this is just the kinetics to show the kinetics of binding using BIA core experiment. It's a very powerful technique again, and you can get the entire kinetic information, including thermodynamics. But only thing I'm concerned as of now is to find out the KD. It binds about 0.4 micromolar KD. And the stoichiometry was important to look for the structure of the protein. And I believe that before I kick from here, I should shorten. So trust me, I'm not going to explain all the data here, but what I'm going to say is that yes, it does. When you make the stoichiometry complex of AF1 TBP, there is a structural change, and in fact, AF1 adopts the structure, and it saturates at 1:1 complex formation. And this is a circular dichroism experiment. And this is just a control to show that it does not happen in the TBP, it's happening just in the AF1. So the question is, if so, what is the functional significance of this? And does AF1 TBP structure formation attracts SRC1, the coactivator we are interested? And the answer is yes. So when you have AF1 bound with TBP and you use the nuclear extract, HeLa nuclear extract, we found that in fact SRC1, this is immunoprecipitation assay using SRC1 as a precipitating antibody and GR as the immunoblotting antibody, that in fact the SRC1 interaction is gone significantly. That means AF1 and TBP interaction is facilitating SRC1's interaction. And in fact, this is the promoter-reporter assay to show that in fact both TBP and SRC1 are synergizing as far as GR activity is concerned. So that everything indicates that in fact AF1 TBP SRC1 complex is involved in giving GR activity. So the first question to ask was that what is the expression of glucocorticoid receptor in these breast cancer cells? And as you can see here, this is a nuclear glucocorticoid receptor without ligand. Mostly receptor is expressed not in the nucleus, but when you give a ligand, dexamethasone in this case, in MCF7 cells, it locates to the nucleus. But look here at MDA, MB231 cells, which are triple-negative breast cancer cells. In fact, without even giving a ligand, GR is active. That means it's constitutively active, and that shows that AF1 activity may be very, very critical in this case. And then we look for TBP and SRC1, these two proteins, if they are highly expressed too, and the answer is yes. And in fact, this is, I'll skip this, and I'll just show this, that in fact, cell viability is very high of the MDA cells when you treat with dexamethasone. And this, I'll skip this, and I'll just thank the collaborators. These are the people who work in my lab, and rest are our collaborators. Thank you very much.
Thank you very much for that very interesting talk. I call on Peter Whiteman now to present the certificate to the keynote speaker.